Hello everyone. Today we have the next exam of our lesson, cell divisions, mitosis and meiosis. Let's start. Today we have the three tasks of our lesson. First, to study the features of cell division and its classification. Second, to compare and identify the similarities and differences between mitosis and meiosis as cell divisions. And last, to formalize the conclusion. I hope you remember information from lecture about cell cycle. Cell cycle is existence of a cell from division to the next division or death. The duration of the cell cycle in the multiplicated cells is 10 or 14, 15 hours and depends on the type of cells, their age, hormonal balance of the organisms, temperature, and other factors. The details of the cell cycle vary among different organisms. The normal cell cycle consists of two major stages. The first is interphase, during which the cell lives and grows larger. The second is mitotic phase. Okay, what is mitosis? Mitosis is a process whereby a somatic cell divides to produce two daughter cells with identical genetic endowment. It involves both division of the nucleus, karyokinesis, and division of the cytoplasm, cytokinesis. Mitosis itself consists of four sequential stages, each characterizing particular aspects of this continuous process: prophase, metaphase, anaphase, and telophase. The first stage is prophase, which is characterized by the following events. Each central pair migrates to opposite poles of the cell. Around each pair, microtubules start growing outward in a radiating pattern called an aster. Microtubules between the two asters continue to grow, pushing the central pairs apart and eventually form the mitotic spindle. The nuclear envelope fragments, the nucleolus disappears, and the chromosomes start to condense. Next stage, metaphase. During this stage, the chromosomes finish condensing and become attached to chromosomal microtubulus. These microtubulus attach to the centromere region at specific sites called kinetochores. Each chromatid is attached to a group of microtubulus from the corresponding pole. Traction on the microtubulus aligns the microchromosomes over the equatorial plane of the cell or metaphase plate, so that their lone axes are perpendicular to the spindle fibers. Next stage, anaphase. During anaphase, the chromatids separate at the centromere and are moved to opposite poles. At this time, they are referred to as daughter chromosomes, each of which is an exact genetic duplicate of the other. This movement is accomplished through the action of molecular motors, sliding along the chromosomal microtubulus toward the mitosis. Telophase is the final stage. During this stage, a cleavage furrow forms around the middle of the cell and constricts, eventually cleaving the cell in two. This is due to contraction of a ring-shaped band of microfilaments associated with the inner side of the cell membrane. The chromosomes decondense, and the nuclear envelope and nucleolus reform, thereby restoring the cell to its interphase state. In contrast to the division of somatic cells discussed earlier, another form of cell division, termed meiosis, involves germ cells. Meiosis consists of two divisions. In the first division, chromosomes form each homologous pair are separated to form two haploid daughter cells. This is therefore termed a reductional division, because the number of chromosomes is reduced by half. In the second division, chromatids are separated from the intact chromosomes, as in mitosis. This is therefore termed an equational division, since no further reduction in chromosome number occurs. The net results in the formation of four haploid daughter cells. Let's continue the first meiotic division. The first meiotic division consists of the following stages. Prophase 1, which is subdivided into 5 periods: aleptoten, zygoten, pachyten, diploten, diakinesis. The first chromosomes each consist of 2 chromatids, begin to condense and become visible as spindle threads. The chromosomes show braid-like thickenings, called chromosomes along their length. Homologous chromosomes pair up alongside each other and form a bivalent in zygoten. This process is called synapsis. The alignment between the 2 chromosomes is very precious and involves the formation of a special structure called a synaptonemal complex that is visible in EM. In pachyten, the chromosomes in the bivalent continue to condense and thicken. Individual chromatids become visible. During this time, adjacent chromatids within a bivalent exchange segments in a process called crossing over. In this way, genetic material of paternal and maternal origin is recombined. In diploten, the chromosomes of the bivalent begin to pull apart, revealing chiasmata, which are the visible manifestations of the crossing over started during pachyten. At the last diakinesis, the chromosomes reach their maximal condensation. Due to partial separation of the homologous during this period, the bivalents exhibit various ring and cross configurations. The nuclear envelope and nucleoli disappear. The next stage, metaphase 1. The bivalents align along the equatorial plane or random assortment. In anaphase 1, the homologous separate and move to opposite ends of the cell, segregation. In telophase 1, the chromosomes decondense and the nuclear envelope and nucleoli are formed. This is then followed by a brief interphase similar to what of mitosis, except that there is no replication of DNA. We now have 2 haploid daughter cells. Remember that the chromosomes in these cells consist of 2 chromatids connected at the centromere. The second meiotic division is essentially the same as mitosis. Prophase 2, where the chromosomes are condensed. Metaphase 2, where the chromosomes align on the equatorial plane. Anaphase 2, where the chromatids separate at the centromere and move to opposite poles. Telophase 2, where the daughter chromosomes, each consisting of a single fiber, decondense. Let's watch a movie. You can also watch the videos and movies on the Ameba Sisters on YouTube page, and you can link to the source.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term, gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes as the newly duplicated portion is still attached at the centromere region, but there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we give the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible. We say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away, the chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually the complete opposite ends, and new nuclei are forming on each side to make 2 new cells. And they are starting to surround the chromosomes on both sides, as this will eventually form 2 cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with 2 identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells. Gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, I hope you enjoy with this film, and right now you have the next task. You need to build a Venn diagram for cell division. Find similarities and differences between mitosis and meiosis. You can also fill out diagrams on the Silver Life platform, and you can link to the source. Thanks for your attention.